With that, I want to introduce our keynote speaker. Dr. Tamara Hershey is the James S. McDonnell Professor of Cognitive Neuroscience in the Departments of Psychiatry and Radiology at Washington University School of Medicine. Her research is in the fields of cognitive and clinical neuroscience, and she's been supported by numerous foundation and NIH award. Her lab uses a range of neuroimaging and pharmacological and cognitive techniques to understand the impact of metabolic and neurodegenerative conditions on the brain across the lifespan. She's particularly interested in the neurodevelopmental and neurodegenerative impact of rare monogenic diabetes, Wolfram Syndrome, and she's going to talk with us today about the value of natural history studies like the one that we're launching with Dr. Amy, and how they've applied that for the Wolfram Syndrome, maybe some lessons that we can take away as a community. Thank you very much.
Thank you. Wonderful. Thank you so much for inviting me, the Bow Foundation and Amy. It's really a pleasure to be here, and it's kind of a nice kind of circle of life or circle of science in that the very first scientific session we had for Wolfram Syndrome, we invited a researcher that studies Niemann-Pick's, another rare neurodegenerative condition in children, to tell us what his experience has been like going from basic science to clinical translational work and clinical trials. And so we had him when he was first starting, and I'm really delighted that I got to play that role for your really phenomenal symposium here and the launch of the natural history studies. It's really very impressive what you guys have put together in such a short period of time. I think people in the rare disease community can learn from each other, and I've already learned things by hearing how you guys are approaching your issues, and the idea here is that maybe you can take some of our experience and help that, use it to help your own progress. I'm also aware that I'm standing between you and lunch, so I will try to be concise. Okay, so I just want to very briefly talk about Wolfram Syndrome just so you understand what we're dealing with. But this is really not a talk about Wolfram Syndrome, it's a talk about what we've learned from doing rare disease natural history studies. So Wolfram Syndrome used to have a name DIDMODE, probably the worst acronym, maybe championing, challenging GNAO1, but it stood for the primary symptoms they thought were necessary to have this condition that they found it described in, I think, 38 by Dr. Wolfram. That all the patients that he saw had diabetes insipidus, which is difficulty regulating urinary output, diabetes mellitus, so it was an insulin-dependent diabetes, optic nerve atrophy, so the children had vision loss that increased over time, and deafness. And it is thought to be pretty rare, 1 in 100,000 in the U.S. to 1 in almost 1 million in the U.K. And it's genetic. It's autosomal recessive. Primary gene is WFS1. There are a small number of families with WFS2. That gene was discovered here at WashU, described in 1998 by Dr. Alan Perma. The diagnosis typically occurs in childhood, and life-limiting complications can occur. So we got launched on a natural history study probably around 2010. So I said the gene was discovered in 98, and it took us that long to do the first natural history study. So you guys are you're winning this race, moving so quickly. It's really inspiring. The delay there was because the person that discovered the gene really didn't do clinical work. So he was very busy describing the gene, the protein it encodes, and what it does in the body, particularly focusing on the beta cells in the pancreas, which make insulin. Since it's a monogenic diabetes, it got a lot of attention from endocrinologists and not so much from people that are specialists in other aspects of the body, like the brain. So it took that long, but pressure from families caused him to set up a registry. You've already done that, so check. And then from that registry, we invited 10 families to come to St. Louis in 2010 and evaluated them. So now we're about to do our 9th consecutive one this summer in July. So why do a natural history study? I think Amy pointed out a lot of the main reasons, but I'll just say that it is the FDA recommended first step towards clinical trials. You have to know what the natural progression is of a disease before you can really tell whether treatments are changing its trajectory in a significant way. I think it's also helpful in trying to understand the full breadth of symptoms, maybe revealing opportunities for treatment that we didn't know about, and giving some suggestion about what the underlying mechanisms might be that can then be tested in iPSCs and animal models, another really important connection to this work. So what we've done over the last 9 years is study genetically confirmed Wolfram patients, not the symptomatically confirmed patients, but genetically confirmed patients. Ages range from 5 to 26 when we started to see them. We've studied 40 unique patients and studied them annually over time. Some will be seen for their 9th time this summer. We also were funded by NIH to continue this work after the first clinic, and were able to also enroll control groups so that we could really understand how Wolfram patients diverge from normal development. And because they have an insulin-dependent diabetes, we wanted to make sure that what we see in Wolfram syndrome was not related specifically to their diabetes, but the underlying neurophysiology of the disease. So we've also done that kind of work. And we do a really wide range of annual assessments because there is a really wide range of symptoms. So our primary focus is neuroimaging, neuropsychology, neurological tests. Amy has been the physician, neurologist, movement disorders expert in the Wolfram clinics for many years. But we also want to look at ophthalmology, audiology, psychiatry, endocrinology, urology, etc. So our clinics take a little longer to collect all of the data because of that. So the first lesson that we learned from doing all this is that you can't go on what was in the literature before there was a gene. So when you look at the literature of Wolfram syndrome, the DID mode kind of syndrome, it suggested that the patients were kind of homogeneous and very severe. But once we have the gene and we can, the people are asking for genetic testing, or their physicians are forwarding them for genetic testing with just a small amount of symptoms, we find that the range of symptoms is much wider than we thought. A lot more heterogeneity. And so, in fact, out of our 40 patients, you can see only 38% fit the actual criteria of the clinical set of symptoms. So that was very interesting, and now the field of Wolfram syndrome research are really trying to rename Wolfram syndrome. Yes, but they're going in the opposite direction. They're referring to it as WFS1-related disorder. And saving Wolfram syndrome for those patients that actually fit this full clinical criteria. So I thought that was kind of interesting. So the other thing we've learned is that Wolfram is much more diverse and has a greater central nervous system involvement than originally thought. As I said, it was really got a lot of attention from pediatric endocrinologists because it's a monogenic diabetes, and so there was a lot of focus on exactly what their diabetes looked like and so on. But as soon as you start studying them and seeing patients and interacting with them and having the right specialists there, you realize that their range of neurologic-based symptoms is much wider, much more profound, much more life-threatening, and affects their quality of life a lot more than their diabetes. And we have shown that by doing quality of life surveys and relating that to symptoms. It's not the diabetes that's a problem. The third lesson is that you really do need that disease-specific rating scale, especially as you realize the variety of symptoms. You want to be able to talk about your patients in a holistic way and be able to compare to, say, another study that's being done in another country, having the same uniform ability to describe the patients and where they're at in general is really, really helpful. So Amy actually helped us with the developing the Wolfram United Rating Scale, or the WERS, where we did reliability, test-retest reliability. Can multiple doctors use this and still get the same answer on the same patients? So that was just an important groundwork. We use this rating scale in all of our studies, and now there's a large European-based clinical trial underway, and they're also using this rating scale there. So again, that will really help researchers and physicians talk to each other about their patients. The other thing that we realized from studying people over time and really looking in depth at their symptoms that we didn't know before is that there were earlier brain abnormalities than we expected. So we thought it was really just a degenerative condition that is, when they start showing symptoms, then things get worse, and that there was brain degeneration going on. But we actually don't know when a lot of our symptoms are starting, because when we study them quantitatively, we can see the differences are there, even at very, very young ages. We've had a 4-year-old tested, a 5-year-old tested, and they have the brain features of older patients, even though they may not have all of the symptoms yet. So that's raised a lot of interesting and important questions about neurodevelopment versus neurodegeneration, and when in the course of the disease those things happen. And I'm just listing here all of the symptoms we've looked at and found abnormalities in relatively early. But kind of in parallel with what Amy said earlier about GNAO1, we see some preservation of higher order thinking in our patients, which is really interesting and may relate to what we see in the brain, and I'll show you that in a little bit. So originally, people thought Wolfram Syndrome, as I said, was really neurologic problems occurred late in the disorder, and that diabetes was early. Then you got optic nerve atrophy, then this condition called diabetes insipidus, and only then did you have brain problems. But because of the studying people very carefully over time, we really can change this model, and we now know that these symptoms are starting earlier. Okay, next lesson, since I am a neuroimager, and we did a lot of neuroimaging in our studies over time, I think we've been able to show that longitudinal neuroimaging can suggest objective surrogate biomarkers for change for the neuropathology in the disease. So I know you have a lot of challenges with GNAO1 in terms of doing MRI scans, but this just shows you the kind of data that you can get. So what I want to show you here is looking at this middle row here. What you see in colors are areas of the brain that in kids without Wolfram Syndrome are changing. So in yellow are areas of the brain that are growing over time and age. And these are, in yellow, are these areas of white matter. This is the insulation of axons in the brain. And we know that normally kids lay down more of this myelin over time. It's a way of, building the strength of those connections. So that's all in yellow. And then in blue are regions that shrink over normal development. That's a very well-known normal phenomenon of synaptic pruning, perhaps, that you're kind of honing in, and the cortex actually does get smaller in childhood. Now, I want to show you what happens in Wolfram Syndrome. So here, what you can see is that you only see blue. Okay? So areas are only getting smaller. Some of that is normal, right, in the cortex. So they're doing normal developmental processes, perhaps, in the cortex, which is where higher order cognition is mostly happening, which may explain why their cognitive functions seem fairly intact. But then what you should also notice is that there's no yellow. There are no areas that are building in volume over time and age. And these are all the white matter areas. So we don't see them laying down this myelin, this protection of their axons over time. That was not known before we did this. And that's really important information if you want to have, say, a good animal model that you can then test hypotheses in and test drugs. You need to make sure the animal model shares this feature. So we have looked at the individual regions and then looked at their volume and how they change. So in normal kids, here are the brain regions that get bigger, so we can quantify how much they grow over time every year. And then, so these are the white matter areas here. And then in some gray matters, it's either a little bit getting smaller or not changing very much at all. And then here they are, all those same regions in Wolfram, and you can see that in the white matter regions, they're all stable or actually losing volume. So very, very different processes going on. And then we found a few gray matter regions like the thalamus that was really surprising. It actually has the biggest amount of decrease on an annual basis in Wolfram Syndrome. It really stands out, and in controls it doesn't really change over time. So that may be a very good marker of a very abnormal degenerative process. So we can then ask, well, does that have any relationship to their symptoms? And what we've found so far is that the more severe they are overall, using that rating scale, so down here on the bottom, this is higher scores mean they're worse overall, all the symptoms together. They're at a sharper decline, abnormal decline in thalamic volume. Okay, so those correlated fairly well, suggesting that it may be a relevant marker of change. And then we can do power analyses to say, well, if you did a clinical study and thalamic volume was your outcome measure, how many patients would you need in your study in order to detect that your drug or other intervention slowed that change? And so we were able to play around with those numbers and suggest that this would be a feasible outcome measure, and that you don't need a huge number of patients in order to detect a difference in the rate of change. And with a rare disease, that's really, really important. We cannot do studies of hundreds of patients. So most studies of drugs in common disorders, Parkinson's, Alzheimer's, they have hundreds and sometimes thousands of patients in order to determine if something works. We do not have that luxury, and so we're looking for really big signals, and this thalamic volume may be one of those. And as I said, we're collaborating with this UK-based European clinical trial. Thalamic volume will be one of their secondary outcome measures. Primary outcome measure in Wolfram is vision, loss of vision over time, and I think the thalamic volume rivals that in terms of its strength. Okay, next lesson is that the neuroimaging patterns that we see, and I already kind of alluded to this, can reveal novel pathophysiologic patterns and support new hypotheses. So I said that the white matter wasn't changing much in the brain of Wolfram patients, so it doesn't seem like they're laying down myelin on their axons. And we can look at this in a different way using a different kind of neuroimaging tool that actually measures myelin. Or infers myelin from its measurements. And what we see in Wolfram syndrome is that they really have less myelin overall compared to control. So a little more specific than looking at volume. So it just sort of confirmed what we saw. And then within one part of the brain, this is the optic radiation, so it's part of the visual system, we saw that they had evidence of hypomyelination throughout the whole tract, but then in a smaller area, they also had evidence of axonal damage, and then an even smaller subset of that area looked like it had axonal loss based on our measurements. And that suggests a potential progress of pathology in Wolfram syndrome, where you might not have myelin protecting your axons, and then over time those axons can get damaged and then die off. And this is a progress that happens in multiple sclerosis and other diseases that have lack of myelination. So we can learn and we can take information from the MS literature or from these other developmental hypomyelination disorders and perhaps use that information in looking for treatments for Wolfram syndrome. So basically, I've said this a couple different ways, but these natural history studies can really lead to hypothesis testing, future hypothesis testing, and more effective clinical trials. So we're using all the information that I've collected in this natural history study over the last 8 years has been used to, actually the collaborator in the UK used our data to get money from the European Union to fund the clinical trial that's just started last month. And so we worked together in that way, but he wouldn't have been able to get that grant without showing the natural history and showing how vision changes over time, showing that MRI measures are useful. So it was very synergistic and really, really helpful for him that we had started doing that so early. By pooling data across sites, we can then look at some of the genotype-phenotype issues that were alluded to earlier. So are there some mutations that are worse or faster progressing than others? We don't really know that yet in Wolfram syndrome, so you guys are really advanced in understanding that. I think neuroimaging is a great tool to bridge between animal models and humans. So if we have an understanding of what is there in the human form of the disease, we can look in the animal models, and if those are similar, then we know we're on the right track with the animal models. It's just a nice reciprocal kind of interaction. And then I really think what you've done here of bringing together people that are studying cells, animal models, patient-oriented outcomes, all of that is really important. All of that feedback and interactions between those kinds of levels of expertise are so important for these rare diseases. And you've already shown it's making the progress go much, much faster than it would have otherwise. So that was basically what I wanted to tell you about. I just want to acknowledge all the people. It's a huge, huge effort to do something like this. We have experts from all different kinds of clinical specialties, from different research specialties over the years, and my lab, of course, and Samantha Rank, who is our clinical coordinator in my lab and helped out with the GNAO1 natural history study this week, because so much of this is logistics and coordination, and that we definitely translates across disorders. And then I also want to thank the funding for all of this. So NICHD, this is an NIH institute, recognizes the importance of studying rare diseases and recognizes the importance of natural history studies. And so they have been very generous and has funded me since 2012. We just got refunded for another 5 years to do all this work. But it's been supported in its early stages by our family foundation, the Snow Foundation, and other small groups. And that was it. So thank you very much.